Okay, I think I've got that fixed. Can you all hear me now? Yes, okay, good. Okay. Well, let's see what it says here. Okay. Yes, yes. Can everybody, okay, does everybody hear me? Okay. Gee, and I sounded so good too. Okay, let me start this over then. And okay. Oh, that's okay. Thank you, though. So I wonder why everybody was so quiet. So let's go ahead and get started now again. Okay, so we're going to talk about, this time you probably won't be able to see the slides. Can everybody see the slides and hear me? I hope so. Can everybody see the slides? Good. That's all I need to know. Thank you. Thank you very much. So, okay, so now we're going to go on. Okay. Peripheral nervous system. We're talking about the peripheral nervous system. Peripheral nervous system is a component of the central nervous system, and it's a separate system. It's everything that's not in the brain or the spinal cord. It has all the spinal nerves feeding into the spinal cord. It has all the cranial nerves feeding into the brain. It has all the cranial nerves coming out and the spinal nerves coming out. Peripheral nervous system has a sensory side and a motor side. Sensory input, motor output. The motor side is subdivided into both the somatic side, which deals with skeletal muscle, and the autonomic side, which deals with smooth muscle and cardiac muscle and the various glands. So on the somatic side, we have control of skeletal muscle. We do not have control of the muscles and glands on the autonomic side. That happens automatically. So we only control the somatic or skeletal muscle side. The autonomic nervous system then is further divided into both the sympathetic division, which is what we call the fight or flight side, or the parasympathetic side, which is our rest and digest and defecate side. The parasympathetic nervous system or parasympathetic division is the dominant side in our bodies. Yeah. Sensory receptors that feed information into the peripheral nervous system and ultimately into the brain and spinal cord have to generate graded potentials that hit threshold. We know all about threshold from discussion in chapters 11 and 12, plus all about threshold with muscle cells. We know that a graded potential is a localized depolarization. When a receptor is stimulated, the stimulation, whatever the stimulus is, will cause a localized depolarization. If that depolarization can hit the threshold at the axon hillock of the neuron that's being stimulated, then we'll get an action potential, and the action potential will run to the next neuron or directly into the central nervous system. That gives us sensation. But we have to have perception of that sensation also, because perception allows us to understand what it is. It's not enough to send an action potential to the spinal cord or to the brain, because all the action potentials look exactly the same. They're always the same. It's what sensory receptor is generating the graded potential that becomes an action potential, and where does it go? Where does it go into the central nervous system? Because that allows us to have perception. An action potential from the retina is the same as an action potential from your skin. But where they get, where they are received and how they are processed gives us perception. It gives us awareness of what the stimulus is. So we have to know, we have to be able to respond to us, we have to be able to detect a stimulus, that's sensation, and we have to understand what it means, that's perception. Now, we have a variety of receptors scattered around our body. We classify them a number of ways. We classify them by the type of stimulus, by their location in the body, and just how complex their structures are. Some of our sensory receptors are very sophisticated, and some are just free nerve endings. In other words, they're just a dendrite. So, stimulus type, mechanoreceptors. They are distorted by some stimulus, by pressure, by touch, by vibration, by stretch. We find mechanoreceptors in our inner ear. They vibrate and give us sound. They generate the vibrations of the hairs inside our inner ear generate graded potentials that may hit thresholds so we can detect sounds. The vibrations in the semicircular canals tell us, if they hit threshold, tell us where we are in space, and at least what plane of orientation we're in. Are we upright? Are we leaning to the left or the right or forward or back? Or are we moving around in circles? We detect blood pressure in our largest arteries of our body by the pressure exerted by the force of the blood going through the vessel, because we're distorting the shape of the artery. Again, we're dealing with pressure. We can tell when we're touched. When someone touches us, we can detect that because we are distorting the mechanoreceptors in our skin. We also, and so they're both internal and external, these mechanoreceptors. Thermoreceptors are internal and external. They respond to changes in temperature. We have two sets of thermoreceptors, one that ranges in hot and heat, and one that ranges in cold. Our photoreceptors are located only in our retinas of our eyes. They respond to light rays, striking the retina, generating graded potentials that become action potentials that go to our occipital lobe, where they get processed so we can perceive what we're seeing. Remember, the photoreceptors in the retina have no clue what they're receiving. They just generate a graded potential that becomes an action potential, and then our occipital lobe, the visual cortex, and the visual association area have to tell us just what it is we're seeing. Chemoreceptors monitor chemical changes. Our ability to smell or taste, for example, depend on these chemical receptors. Our blood pH, the acidity of our blood, the salt concentration of our blood depends on these chemoreceptors. So they work both internally and externally. And then, of course, we have our nociceptors, our pain detecting receptors. They respond to changes in temperature, changes in pressure, exposure to acids or bases on our skin, exposure to trauma. And we can have pain receptors both, again, internally and externally. So location-wise, we have the exteroceptors. The exteroceptors monitor the external environment, anything outside the body, touch, pressure, pain, temperature, even vision, sound. All these are exteroceptors. They are telling us what's going on outside of us. Interoceptors are in our viscera. They monitor changes in temperature and pressure in our blood vessels. in our small intestine. They monitor chemical changes in the blood, stretch of the various organs. Usually, we know when our bladder is full because it stretches. We know when it's time to defecate because the rectum is full and it's swelling. We know when we're thirsty because our salt concentration goes up a little bit in our plasma. These are all internal changes. The interoceptors are detecting all this. So, and telling us when they generate an action potential, they're telling us what's going on inside. Perhaps we have a pain in our lower right quadrant, or we have a pain in our upper right quadrant. What is that telling us? Well, first we have to perceive the pain, and then we have to interpret what it means. So, of course, sometimes a pain is just a pain also. Proprioceptors tell us our position in space, where we are. Our muscles and our tendons and our joints have proprioceptors telling us where we are, telling us what kind of movements we're exerting. Are we exerting? Are we staying within the limits of our muscles and our arms and legs, or are we going past the limits in here? These are the proprioceptors. Structure-wise, they're either simple or complex. The simple receptors give us all the general senses: the touch, the pressure, the pain, temperature, things like that. The general senses are the complex receptors. The special senses, the general senses are just usually a dendrite at the end of a neuron, at the afferent end of a neuron. It's either encapsulated or it's not. It's either encapsulated or it's free. We call that just the free nerve ending, which is really just a dendrite. And the dendrite is going to be picking up touch or pressure or pain. Now, if we have the dendrite covered with connective tissue, we say it's encapsulated. We generally have those deeper in the skin to detect deep pressure, for example. The special senses are all housed in their own very sophisticated sensory organs, like the eye or the ear or the nose. And they are very complex. So we have vision, and we have taste, we have balance, we have odor, we have vision, taste, odor, balance, whatever. So hearing, yes, I keep forgetting that one. Okay, so all housed in our very sophisticated sense organs located in our head. Now, a lot of our sensory input, well, actually most of our sensory input comes into the somatosensory cortex in the cerebral cortex. The somatosensory cortex is found in the post-central gyrus, right behind the central sulcus. We get our input from skin and from the viscera and from our proprioceptors. So we have the exteroceptors detecting changes and stimulus all over the surface of our body. We have the interoceptors detecting changes and stimulus internally in the viscera, and the proprioceptors are telling us where our arms and legs and hands and feet and muscles are, where they are and what they're doing. We send that information in through the thalamus to the somatosensory cortex, and then the somatosensory association area behind the cortex is processing all this information for us so that we can figure out just what is going on. All the inputs coming in from all these sensory receptors throughout our bodies. We process it as it goes along, and once we get, we eventually can get it to the central nervous system so that we can determine what's happening. So we have our perception of the stimulus and what it means. So now one of the problems we have, and it's not really a, it's sort of a good problem sometimes, is that this sensory input that comes in adapts. If we have a constant stimulus going on, in many cases, we will adapt to that stimulus. Background noise fades away because we no longer pay attention to it. Reticular activating system sort of tunes it out. The sound of the heating and cooling system, the air movement from the air handlers, the pressure of the chair on your back, the tag in the back of your shirt, if you have a tag, rubbing against your neck, the pressure of our clothing on our skin. We may not be paying attention to it. It doesn't, we no longer respond to it. Background noise, if you live near a major road. If you live near the interstate, you may not hear the noise of the traffic. Just like someone that lives near the ocean no longer hears the sound of the surf hitting the beaches. Whereas when you go there on a vacation, well, that's the first thing you hear. And everybody says, "Oh, I can really sleep well because I hear all the sound of the ocean, of the surf." But people that live there, it fades in the background because they've been exposed to it and exposed to it and exposed to it and exposed to it. In fact, it just, the only time that you would hear it at the ocean is if something extraordinary was going on. The only time if you live near the interstate, the only time you would notice something is if the sound stopped. The sound of the traffic stopped. If the road becomes blocked because of a big accident somewhere, a mile out of the way, and you don't hear the noise of the road anymore, then you detect it. Otherwise, it just sort of fades in the background. So, adaptation, we adapt to a constant stimulus. We no longer become as sensitive, we stop generating graded potentials, and we may end up stop sending action potentials. Many types of sensory receptors adapt very quickly. Touch receptors, pressure receptors, odor. We get used to odors very easily. These are what we call phasic receptors. They adapt very quickly to a stimulus. They adapt very quickly to a stimulus. It doesn't take much for them to get bored with the stimulus, if you will. So, odors in particular, you get used to the odor. We say, well, I'm used to that odor. I don't notice that odor. The tonic receptors don't adapt. On the other hand, phasic receptors do, tonic receptors don't. They adapt slowly, they may not adapt at all. Pain receptors never adapt to pain. They're always telling you that pain's there. Proprioceptors always tell you your position in space. They don't adapt because we're always moving. We're always moving around, so they can't adapt. Just like pain receptors, pain is our body's way of saying, stop doing that, it hurts. We don't want to adapt to it. We want to be constantly reminded that something is painful. So phasic receptors adapt, tonic receptors don't. Pressure, touch, and odor adapt. Pain and proprioception don't. So now when we receive these signals, if we're not adapting, what will happen? We have 3 levels of processing. We start out at the receptor level. The free nerve ending, for example, is going to be stimulated by something. Pain receptor, temperature receptor, whatever. It will generate a graded potential that if it hits threshold at the axon hillock will trigger an action potential. That's the first thing we see. We have the receptor there sending, we hit threshold and we're sending it up into the ascending tract, the white matter of the spinal cord. We continue to synapse. we get up to the circuit level where we get to the thalamus, and we continue to process this information, and the thalamus then routes it out to the area of the somatosensory cortex that will tell us what's going on. That gives us so the last level is the perception level. When we get to the cortex, the cerebral cortex and the somatosensory cortex tells us what's going on. That's where perception occurs. Perception doesn't occur anywhere else except in the cortex. We have to send it, we have to send the signal and massage the signal, but we're not going to interpret it until we get to the cortex. So for us to generate, to be able to perceive something, we have to have a sensation that triggers an action potential. And we have to have the right receptor for the right stimulus. You can't hold your hand out and detect light. No photoreceptors in the palm of your hand. Plus, you have to hit the receptive field. The stimulus has to, the stimulus has to hit the receptor. You know, we don't have this uniform blanket coverage on our bodies. We have receptors all over the place, but if we miss a receptor, we may miss the signal. So we have to have the receptive field. And if we do have the receptor in place and we do have a stimulus, then again, we have to have what we call transduction. We have to create a graded potential from the stimulus, has to create a graded potential that is sufficiently strong enough to hit threshold to generate the action potential. This is everything that has to happen at the receptor level. If we don't hit threshold, we don't get an action potential. The second level up, we process. We send our signal up to a series of sensory neurons feeding into the spot to the spinal cord, and then ultimately through the thalamus into the processing level, into the cortex. First order, second order, and third order neurons will ultimately get us to the somatosensory cortex. We send that signal, we have, you know, and once we've hit threshold, we're going to get the action potential running to each neuron, and that will get us to the thalamus. But again, perception doesn't take place until we get to the cortex, to the somatosensory cortex. And perception lets us detect that there was a stimulus, something is touching our skin. It tells us how strong the stimulus is, and it tells us where it's coming from. So we have a stimulus, we know how strong the stimulus is, and we know where the stimulus is hitting our skin. You know, while all action potentials are alike, we'll talk about the intensity of the stimulus. We know that the more rapidly we receive an action potential, the more intense the stimulus is. So we detect the stimulus, we know how strong it is, and we know where it is. That's perception. That is the processing in the somatosensory cortex. All of our sensory input depends on this. We have to have a sensory receptor that is stimulated, generates a graded potential that is strong enough to have an action potential that will trigger first, second, and third order neurons to each have an action potential, getting us to the thalamus, ultimately to the cortex. Then at that point, then we have perception. We detect the stimulus, we know how strong it is, and we know where it's coming from. At the level of perception in the cortex, we can identify the specifics of the stimulus. Is it pain? Is it pressure? Is it temperature? Is it an odor? Is it a taste? We can identify what kind of stimulus it exactly is. Because all action potentials look alike, but we can tell by processing where they're being received just what we're dealing with. That's the feature abstraction. We also have what's known as quality discrimination. That allows us to identify the various submodalities of the sensation. We can detect various odors. Here, you can detect the odor of different flavors in a food. You can detect the different flavors themselves. We can detect, if you're eating a chocolate chip cookie, you can detect the flavors of the chocolate and the butter and the cookie itself. Or perhaps you're eating, it says pepperoni pizza or cheese pizza. Maybe you're eating, your favorite Mexican food or Chinese food, or you're having sweet and sour shrimp, for example, or sweet and sour pork, or you are eating key lime pie. Quality discrimination is really good on foods. For example, key lime pie, you detect the graham crackers and the whipped topping or the meringue topping on the surface. You detect the subtle odors and subtle flavors of the key lime juice in the pie itself. You detect the textures of the filling of the pie. You detect the textures of the crust. So this is all quality discrimination. On your clothing, for example, you detect, do the sleeves of your shirt fit well? Do they button properly at the cuffs? If you have cuffs on your, buttons on the end of your sleeve, are they tight? Are they loose? Do they annoy you? Is there a tag in the back of your neck, on the back of your collar? Is that bothering you? These are all different types of sensations. Is the tag there not just a touch irritation, but also a painful irritation? Perhaps it has some little piece of plastic hanging on there. It's digging into your skin. Quality discrimination lets you pick out all the different sub-sensations. It's touch, but it's also painful. It's touch, but it's also cold and painful, or hot and painful. So when you bite into a hot slice of pizza, yes, you get all the flavors of the cheeses and the pepperonis, but it also may hurt if the cheese is really hot. It sticks to the roof of your mouth and it's very, very hot. So there you're getting pain on top of all the flavors too, in addition to the tomato sauce and the cheese and the pepperoni and the crust, and maybe the onions and the garlic and the peppers and whatever else. That's quality discrimination. The components of the sensation, all the different components, in the food or in the odor. You detect odors and you may detect all the variety of subtle odors in there. Pattern recognition, we find patterns in various stimulus. You look at a cloud and say, oh, it looks like a sheep. All clouds look like sheep. But you say, that cloud looks like, a dog, or, that cloud looks like a waterfall or whatever. Or you look for a pattern in some stimulus. You hear a sound and you recognize a pattern to the noises. When you're driving your car, the pattern, the sounds coming from the engine, we don't pay attention to it unless the pattern of the noise becomes irregular, something we're not used to. We listen to music. We recognize melodies because of the patterns that we have become familiar to. We have become very familiar with certain patterns in music. We know what the next note's going to be because we have that stored in our memory. So we recognize various auditory patterns. very easily. Or visual patterns. We recognize those very easily. We know what jars us is if we have a visual pattern where one piece is out of place. You know, where you're putting together, you're putting together a puzzle and one piece is missing. You detect that right away. If you've assembled something and you have a part left over and you notice that, you know, we recognize a pattern. We recognize the absence of a pattern that should be there. So we recognize objects by touch and by shape. We can identify objects without having to look at them, simply by their shape. We can feel what they look like. We can feel their shape and detect what they are. We recognize a fork by its shape, by the tines on the end of the fork. We recognize a knife by its handle and its sharp edge. We recognize various keys by the shape of the key. So, or coins by their shape. The somatosensory area allows us to process all this information, not just features, all the various parts of a sensation, not just the submodalities, not just the patterns, but basically also by their shape itself. You know, you recognize a flashlight, your phone, a phone, any phone. You can detect that right away. You know, if you have a plastic box about 6 inches long and about 3 inches wide, and it's very, very smooth on one side, likely a phone. You know, if you're not looking at it directly, if you're just identifying it by touch, you know, or its shape. You know, we're used to the shape of our phones, of our smartphones. And so we're more than likely to figure out what it is simply by its shape. So, okay, so that deals with the somatosensory input and processing all this information. Again, these three are very important: feature abstraction, quality discrimination, and pattern recognition. You know, very important things to remember. Next time you're eating pizza, think of the quality discrimination. Next time you hear music, think of the pattern recognition. Next time you're putting something together, think of the feature abstraction of it. All the different visual stimulus cues you're getting from the instructions to get the finished product. So, now, what about pain? Pain is our body's way of saying, stop doing that, it hurts. It warns us not to do something. We can stop doing it so we don't have damage. It is our body's way of saying, that's going to hurt, or that already hurts. You step on something sharp and you yank your foot back so that you don't damage it any further. You know, pain can be triggered by pressure, by temperature, by the release of histamines. Because histamines cause swelling, and swelling causes pain. Exposure to acids, for example, can cause pain. The pain action potentials travel along the same neurons that release the excitatory neurotransmitter glutamate, and also substance P. Substance P allows us to mitigate the strength of the signal so that we are still aware of the pain, but it may not hurt as much. So perception of pain. We aren't going to block all the pain. We're still aware of the pain, but at least we can deal with it better. You know, so like the same neurons that are going to release glutamate. Glutamate is an excitatory neurotransmitter, but it's the same neuron maybe releasing substance P, which tends to mitigate, to reduce the effect. We also may be releasing endorphins to counteract the pain. Endorphins are pleasure neurotransmitters that make us feel better. So maybe the pain's still there, but we're not thinking about it as much. We feel better about something else, and so we aren't as sensitive to the pain as we were before. So now, tolerance of pain. We all perceive pain at the same intensity. Action potentials are all the same. Pain receptors, when they're triggered, are going to generate an action potential all the time. They're all going to be of the same intensity. Every action potential is going to be the same as any other action potential. The tolerance to pain is what varies. How sensitive are we to pain has nothing to do with our pain threshold, because our threshold for pain is exactly the same for everybody. When we hit threshold, we get an action potential, we get pain. But it's how well we respond to that threshold. The tolerance means how do we deal with the pain when it arrives is what's going to vary. Individuals have variable pain tolerance. We all hit the same threshold for pain, but our tolerance, our ability to respond to the pain is going to vary a lot by whether or not we're releasing endorphins or whether or not we can use natural, the example over here, substance P to help block some of the signal of the pain. So many people say that they have, they may say they have a high pain threshold. No, everybody's threshold is the same. It's their tolerance to pain that's going to fluctuate. Now, types of pain: visceral pain and referred pain. Visceral pain comes from the viscera, internal organs. Stretching, lack of oxygen to tissues causes pain. We call it ischemia. Pain in the heart from lack of oxygen supply that causes a pain called angina. If it's severe, it becomes a heart attack. Chemical pain, muscle pain. This is all visceral pain that occurs. It's all internal pain. So, we get pain from all sorts of actions in our viscera: swelling and bloating and stretching and irritation. The problem comes into when we deal with referred pain. Referred pain is going to try to throw us a bit of a curve here. Referred pain is pain that appears to be coming from one part of the body when it's really coming from another area. The pain coming from your heart attack is traveling along the same neurons that somatic pain travels. The somatic pain, the somatic fibers from your skin telling you that you've got pain in your left arm are the same fibers that the visceral pain from the heart attack is traveling. In other words, you would mistake heart attack pain for pain in your left arm. Very common occurrence. A person having a heart attack may very easily have pain in their left arm because the pain signals from the heart are traveling along the same nerves that somatic pain in the left arm does, and our perception is, oh, my left arm really hurts, or my left mandible really hurts. Same nerve that innervates the mandible also innervates the heart, and so pain in the lower jaw on the left-hand side is often an indicator of a heart attack. Sometimes it's just a toothache, and sometimes a sore left arm is just a sore left arm. Other times it might be an indicator of something going on with the heart. So very common though to have left arm pain during a heart attack, or left lower jaw, or pain in the abdomen. You know, again, it's the same nerve that carries pain from the heart is also carrying somatic pain from the abdomen, from the arm, and from the mandible. So, okay, so what is a nerve? A nerve is a collection of axons. They can be myelinated axons, they can be non-myelinated axons grouped together. And it is usually lots and lots of axons together. Now, the nerves in our bodies are either going to be spinal nerves or cranial nerves. Spinal nerves originate in the spinal cord, and cranial nerves originate in the brain. So we have two types of nerves. Now, remember, these same axons, if they were in the central nervous system, would be called tracts. But here they're called nerves. In the peripheral nervous system, we call them nerves. The individual neurons are covered by layers of connective tissue. Just like we had the endomysium in muscle cells surrounding each individual muscle cell, here we have the endoneurium. This is a layer of connective tissue that surrounds the individual axon and their myelin sheath here in the peripheral nervous system. The perineurium is connective tissue that surrounds clusters of neurons that are formed into fascicles, just like in muscle cells. We have the fascicles, clusters of neurons together surrounded by the perineurium. And then the entire nerve is surrounded by a layer of connective tissue around all the fascicles called the epineurium. Here we see what it looks like. There's your nerve right here. That's the epineurium on the outside. These are all fascicles here. This is the perineurium surrounding a fascicle, and there's more perineurium there and there. These are all fascicles. And then we have our endoneurium surrounding each individual axon. There's an axon with its myelin sheath around it, and there's the endoneurium surrounding it. Very much like, very similar structure to what we saw in muscle cells. Now over here is an actual photograph of this. Here we see the individual axons right here. We see the endoneurium around each axon. We see the perineurium surrounding this. This whole structure here is a fascicle. So we have the perineurium surrounding that. And there's another fascicle. There's another fascicle and another fascicle. We have the perineurium around each one. And here's the epineurium surrounding the entire nerve. Here we see it in close-up. The endoneurium surrounding each individual axon. There's an axon there and an axon there. This is all part of a fascicle. There's the perineurium here. Somewhere on the edge, we have the epineurium. So we have the three layers, endo, peri, and epi, surrounding the individual axons, fascicles, and nerve. The nerves that we encounter are going to be usually a combination of both afferent and efferent fibers. Sensory comes in on one side of the nerve, motor goes out on the other side of the nerve. And these nerves share, the structure has both a sensory afferent and a motor efferent side to it. Some of these nerves are going to be somatic, going to skeletal muscle. Some are going to be autonomic, going to the viscera, going to the organs, the glands, the cardiac muscle, or the smooth muscle. So we classify these nerves based on which way the signals are going to go. We have mixed nerves that are both sensory and motor. Signals go to and from the central nervous system. If it's sensory only, then we only go to the central nervous system. If we are motor only, we only go away from the central nervous system. So we have sensory, and we have motor, and we have mixed. The cell bodies of our neurons in the peripheral system are called ganglia. Remember, in the central nervous system, we call them nuclei. Here we call them ganglia. And we have various types of ganglia here. We have we have ganglia from the autonomic neurons. We have the visceral and motor ganglia. We have the sensory and somatic ganglia from the dorsal root ganglia. What you need to remember is that the ganglia are the cell bodies in the peripheral nervous system. So, now what about neurons? Can they be repaired? In the central nervous system, the answer is no, at least not right now that we're aware of. If the cell body of a damaged neuron in the peripheral nervous system is left intact, if the cell body has not been damaged, if the axon is the only thing that's been damaged, then the nerve in the peripheral nervous system can regenerate itself. It can regenerate its axon and reconnect the two ends of the damaged axon. If that wasn't the case, then how could we do transplants? We could never transplant an organ without the ability of the peripheral nervous system to regenerate its axons. They grow back together. This does not happen in the central nervous system. In the CNS, the axons don't regenerate. The oligodendrocytes, which are the counterpart to the Schwann cells that generate the myelin, the oligodendrocytes prevent regeneration. The astrocytes are going to cause scar tissue to form at the site of the injury, blocking any repair. Scar tissue doesn't do anything for us anyway. So we may be able to treat that by blocking the role of the astrocytes and blocking the growth inhibitors that are coming out of the oligodendrocytes. Right now, the CNS doesn't repair anything. However, in the peripheral nervous system, it's different. If the cell body is intact in the peripheral nervous system, then we will generate, Schwann cells will form a growth tube around the two ends of the axon, and the axon will regenerate and will grow a new myelin sheath, and the axon will function like as it did before. So we see, let me get back here. So if the axon is cut, then the oligodendrocytes will grow almost like a cast around the site of the injury. And then the ends of the two ends of the axons will start growing together and fuse. And once they're healed up, then they're now as good as new. So we see this all the time. Otherwise, what would be the point of reattaching a body part if we couldn't have nervous control of the body part, or doing a transplant? So if the damage is not great in the peripheral nerve, it can regenerate itself. Okay, now there are 12 cranial nerves in the peripheral nervous system. There are 31 spinal nerves. The 12 pairs of nerves in the brain. Most of them are mixed, meaning both sensory and motor. 2 of them are sensory only. We classify them, or rather we identify them by a number, 1 through 12. And we have the list of the 12 cranial nerves are olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibular, cochlear, glossopharyngeal, vagus, spinal, accessory, and hypoglossal. Now, how do you remember all that? There is a mnemonic. There are numerous mnemonics. Some are very simple, like this one, that are based on the first letter of the name of each nerve. And this is the one that I learned when I was an undergraduate, and many people still learn. On old Olympus towering tops, a Finn and a German viewed some hops. As silly as it sounds, if you learn the mnemonic, you know the order of the 12 cranial nerves. So are they all sensory? Are they all motor? 10 of them are sensory and motor, and most of them are, but 2 of them are sensory only. So now, here are the cranial nerves: olfactory, sensory, optic, sensory, oculomotor, and trochlear are motor. Trigeminal is both, abducens is motor, facial is both, vestibulocochlear is sensory, glossopharyngeal is both, vagus is both, and the last 2 are both motor. So now, what do they do? Well, olfactory is sensory. Extends from the olfactory mucosa in the nasal cavity to the olfactory bulb. And we run the action potentials then run down the olfactory tract to the cortex and the association area. Remember, once we enter the central nervous system, it ceases being a nerve, it becomes a tract. So once we come from the mucosa to the bulb, once we get to the bulb, then we're no longer a nerve. So the optic nerve supplies action potentials from the retina to the visual cortex. Problems with the optic nerve: if we have visual acuity or visual field issues, visual field, we don't, we're not receiving, the photoreceptors there are not functioning. Here, we don't have good visual acuity. Oculomotor is both sensory and motor. It deals with our sense of position, that's proprioception. It also tells us how to move our all 6 of the extrinsic eye muscles. It allows us to accommodate the change, the shape of the lens. The ciliary body is controlled by the oculomotor nerves. Oculomotor nerve, the ciliary body, which causes the lens to accommodate, to change its shape for distance vision or close vision, to constrict the pupil, to restrict light coming in. This nerve controls all that from a motor perspective. Perception-wise, it gives us our sensory proprioception. Where are we? It gives us our sense of position. Trochlear. Proprioception, again, deals with position and motor control of the oblique eye muscles. An individual that has double vision or a strabismus, where we used to say cross-eyed, the oblique eye muscles are pulling the eyes towards the midline. Issues with this is driven by a malfunction of the signal going to the oblique eye muscle. The trigeminal nerve, one of my personal favorites here, 3 branches to it, can detect touch, pain, and temperature on the face. We have the ophthalmic branch, which is near the eyeball. On each side, we have the maxillary branch and the mandibular branch. And sensory input, when we're having dental work done, involves blocking the signals from pain signals in either the maxillary or the mandibular branch of the trigeminal nerve on either side. It comes out in 3, we call it trigeminal because it has 3 distinct parts there. It also stimulates the muscles of mastication. That would be like the temporal and the masseter muscle. Tell us to elevate our lower jaw to chew. The abducens nerve, proprioception, and also our lateral eye muscle. Notice on the in the second image how the eyeballs are pulled in towards the midline a little bit. Lateral motion there. Here we see the facial nerve. It innervates the taste buds on our tongue, that's sensory, but also it controls the facial muscles. And this is Bell's palsy, where we have paralysis of part of the facial muscles here. And so this facial nerve, number 7, is involved with this. The vestibulocochlear nerve gives us both auditory and balance. The cochlear branch is what we used to call our auditory nerve. It sends action potentials to the association area and the auditory cortex, so we can hear things and tell what we're hearing. The vestibular branch gives us our sense of balance in here. Vestibular branch is feeding information from the semicircular canals. The glossopharyngeal nerve, if you've ever had the desire to be a sword swallower, then you can appreciate the glossopharyngeal nerve, number 9. Sensory, it is the last, our taste buds, the last 1/3 of our taste buds are detected by the glossopharyngeal nerve. Motor-wise, it allows us to elevate our throat when we swallow and secrete saliva. So the ability to do something like this is thanks to the glossopharyngeal nerve. The vagus nerve. You'll hear a lot about the vagus nerve in AP2. The vagus nerve gets sensory input from all over the viscera, but it also is used to control the heart rate. It dumps acetylcholine into the heart to slow it down. That's its job. And it's controlled by that's controlled by the medulla oblongata. The vagus nerve can also be carrying signals from the viscera. So pain in the heart, part of the viscera, pain in the heart may be perceived as referred pain somewhere else because of where the vagus nerve runs. You'll hear all about the vagus nerve in AP2. So now, okay. Number 11, the spinal accessory nerve controls the sternocleidomastoid and the trapezius. It has both a cranial portion and the spinal portion. It affects our throat and our ability to, the skeletal muscle in our throat. And of course, it also affects our ability to turn our head. And number 12, the hypoglossal nerve. It has a proprioceptive role, position of our tongue, but it also, from a motor perspective, it tells us to swallow. It tells us how to speak properly. The hypoglossal nerve, if the medulla is damaged in the area controlling the hypoglossal nerve, your patient will frequently aspirate their food or have difficulty speaking or swallowing. If the medulla is damaged, the tongue may actually point to one side, to the damaged side. If the tongue is stuck out, because of the damage, the tongue may deviate, may point over in the direction of the damage. So now there are 31 pairs of spinal nerves, 8 cervical nerves, 12 thoracic nerves, 5 lumbar, 5 sacral, and 1 pair of coccygeal nerves. So all 31 pairs here. Okay, we only have 3 slides to go, so let me finish this and we'll be done with it. Hang on a second, let me get back to where I was. It won't take you long to finish this. We'll be done with chapter 13. Okay. 31 pairs of spinal nerves. Now, every spinal nerve has 2 roots to connect to the spinal cord. We have the ventral root, which is motor. The ventral root comes in on the anterior surface of the spinal cord, the belly side of the body, the stomach side of the body. So that's the ventral root. That is the motor root. It comes out of the ventral horn, going out to skeletal muscle. The dorsal roots come in on the sensory side, on the posterior surface of the spinal cord. So we have this butterfly gray area in the middle, and the area near the root, what we call the horn. Let me show you a picture here. Oops, didn't mean to do that. Let me stop it. Why did it do that for me? Well, I guess we're just going to have to stop and finish these last 3 slides on Tuesday. So it has been a interesting day. Anyway, thank you for your patience. I appreciate that. And I am going to, does anybody have any questions before I get us out of here? We will finish those last 3 slides on the spinal cord on Tuesday. So remember, there are no labs next week either. We're done with lab. Okay, I'm going to get us out, and you all have a good rest of the day.